membrane. Other protein located on the lumen of the ER, they always have one. They always carry one amino acid sequence, and this sequence they style as the KDEL. KDEL, okay. So KDEL is one signal sequence that they indicate the protein will stay on the solute on the ER lumen. So in this image, you see this is the solute protein contain the KDL signal, and this protein they can bind to the KDL receptor on the membrane of the ER. So when the KDL bind to the empty KDL receptor, they also can trigger the transport to the formation of the vesicle by the COP1 coat protein. Okay, so you see here, in the K, okay, so in the right image from the Golgi apparatus, they need to transport the solute protein present as the yellow square. So this yellow square belong to the secretory protein, and they need to be transported to the Golgi apparatus. So this solute protein is concentrated on one part of the ER membrane fed to the Golgi apparatus and trigger the formation of the vesicle here. And also in this part of the ER membrane, they also locate the receptor of the KDL receptor. So during the formation of the vesicle to collect the solute cargo protein, they can also contain the KDL protein in the ER lumen. Also randomly, they can take the KDL protein, the solute KDL protein in the lumen of the ER. And in the other vesicle, they also take the receptor of the KDL on the ER membrane together with the cargo protein to make the vesicle here. So both types of these vesicle will be transported to the ER to the Golgi apparatus. So when after the fusion of the vesicle to the Golgi membrane, so they release both the cargo protein, the zero one, and the KDL solute protein, and also the receptor, KDL receptor, present now on the Golgi membrane. One thing I would like to note here is that the difference in the pH value between the ER and the Golgi apparatus. In the ER, the pH is neutral, but in the Golgi apparatus, the pH is weakly acidic, and the more Golgi apparatus as the cis phase, as the trans phase, the more acidic of the pH value. Why I mention about the pH value? Because the pH value here, they involve to the binding of the KDL protein and their receptor. So as the pH of neutral, there is no binding of the KDL protein and their receptor, or the binding is very weak. But whenever the pH is acidic, so this acidic enrichment induced the binding of the KDL protein to the the receptor. So when the chiral protein and their receptor stay in the Golgi apparatus, the pH of the Golgi apparatus induce the binding of the chiral and their receptor. And this binding will trigger the formation of the vesicle by COP1 protein of the Golgi apparatus and send back the vesicle from the Golgi apparatus to the ER membrane. So when the vesicle fills with the ER membrane, and because of the pH neutron in the ER, so the binding of chiral protein and their receptor is broken and release the chiral protein in the ER lumen. So this is one process. This is the mechanism of retrieval pathway in from the Golgi to the ER. Okay. So the image in the left illustrating about the vesicle formation on the Golgi by the support from the COP1 protein. Just 1 minute. Okay, please. Okay, so in in this example of one type of the protein in the ER, you can see that the first thing that this protein they contain the chiral signal sequence, and this sequence will help the protein to bind to the receptor, and the binding of the protein with the receptor will trigger the formation of the vesicle and transport back the both receptor and the chiral protein to the ER. And the second point here is that the binding of the chiral protein with the receptor only happen when the resist protein stay in the pH acidic pH value, which is present in the Golgi apparatus. For the pH neutral, the neutral pH value, the binding of the chiral protein and their receptor is broken, and they release the chiral protein in the ER lumen. Okay, so that is the retrieval pathway between the Golgi apparatus and the ER. So when the vesicle come from the ER to the Golgi apparatus and fuse with the cis phase of the Golgi network, all the protein will be released into the Golgi lumen or spread on the Golgi membrane. And during the time stay in the Golgi network, the protein also under the modification by many processes inside the Golgi, such as the phosphorylation or the adding of the sugar group, and the other modification. So the protein will be transported from the cis phase of Golgi to the trans phase of the Golgi, and during this transportation, they also they are also modified by some processes in the Golgi, which is you already learned it in the part of the intracellular compartment by Dr. Long. So whenever the protein stay in the trans phase of the Golgi, that means this protein is already modified and ready to be sent to the other organelle, such as the lysosome, the secretory vesicle, or the plasma membrane. So from the Golgi to the other part of the cell, they also have the three type of pathway. The biosynthesis secretory pathway, the endocytic pathway from the out of the plasma membrane to the endosome or lysosome, or the retrievable pathway from the endosome to the Golgi. So here is one example of the transport from the Golgi apparatus to the lysosome organelle. So as you learn at the lesson of intracellular compartment, the lysosome is somehow like the stomach of the cell, where they can digest many types of the molecule inside the cell, because they contain a set of the acid hydrolase, like nuclease, protease, glucosidase, lipase, phosphatase, sulfatase, and phospholipase. So this set of hydrolase can digest many types of the molecules, from proteins, the lipid, the lipids, and glucose molecules in the cell. And these hydrolase, they only work as the acid enrichment. That means the low pH value, about pH 5. And to maintain this acidic enrichment, so the on the membrane of the lysosome, they need to express many H pump, proton pump, to pumping the proton from the cytosol into the lysosome, and this pumping also needs the energy from the ATP hydrolysis. So for the hydrolase enzyme, they need to be synthesized and transport from the ER to the Golgi apparatus and then to the lysosome. And for the mature hydrolase that is spread in the lysosome and work, so they need to link to the mannose 6 phosphate in the software called the M6P, the mannose 6 phosphate. When one phosphate group will be linked to the carbon as the carbon 6 of the mannose sugar. So how the hydrolase enzyme is modified and transported to the lysosome. Okay, so this schema illustrates about the way the hydrolase is transported from the Golgi to the uh, endosome, and during this they also do some modification. Okay, so the protein hydrolase is synthesized on the ER, okay, and then transport from the ER to the cis phase of the Golgi apparatus. So at the beginning, the hydrolase stay at the precursor. That means the immature hydrolase without function. So the hydrolase precursor from the ER to come to the cis phase of the Golgi will be added a phosphate group and to make the mannose 6 phosphate attached to the hydrolase here. So when they come to the 6th phase of Golgi network, the phosphate is attached to the carbon 6 of mannose sugar on the hydrolase. And after that, this precursor is transported from the cis to the trans phase of the Golgi network. At the trans phase of the Golgi network, the the M6P, that means the mannose phosphate, is recognized by their receptor on the membrane of the Golgi apparatus and bind to their receptor. So now the precursor bind to the receptor and trigger the formation of the vesicle. And the type of the vesicle here is the clathrin coated vesicle. So the clathrin coated vesicle is pinched up from the Golgi and be transported to the endosome. After fused with the endosome, because of the pH value of the endosome is more acidic than the Golgi, so the binding of the precursor and the receptor is weakened and release the precursor to the lumen of the endosome. The receptor still stay on the membrane of the endosome and be transported by vesicular transport back to the Golgi to ready for another cycle. For the precursor, when they stay in the endosome, uh, also there is the removal of the phosphate group on the mannose sugar. And at this time, the precursor becomes the major hydrolase and ready to do their function as a digestion enzyme. So in this image, you can see both the biosynthetic secretory pathway and the retrieval pathway. For the biosynthetic secretory pathway, that means the transport of the hydrolase from the ER to the Golgi to the endosome. And for the retrieval pathway, this is the transport of the mannose phosphate receptor from the Golgi to endosome and back to the Golgi network. And in this example, again, you can see that the pH value as a different organelle can trigger or inhibit the binding of the receptor and their cargo. So as you see in the example, the trans phase of the Golgi network when responsible for the exocytosis of the cell. That means many secretory protein will be transported by this way from the trans phase of the Golgi network to the cell exterior through the vesicle formation. So this is an image of one example of exocytosis way in the cell. Upper is a schema illustrate one vesicle contains the cargo, that means the secretory protein, transported to the plasma membrane and fused with the plasma membrane. After the fusion, all the soluble protein will be pushed out of the cell. And maybe they come to some vessel, blood vessel, and transported to the other cell. Upper image is an electron microscopic image illustrate about the two vesicle one vesicle just come close to the plasma membrane, and the other vesicle is already successful fused with the plasma membrane. And the dark clump here is secretory protein and the other molecule, and this molecule will be sent out of the cell. So uh, the transport of the molecule from the transphase of Golgi network will be sorted, and the protein or the other molecule, when they stay at the transphase of Golgi network, they can be either belong to the signal mediated diversion to lysosome, or they belong to the constitutive secretory pathway, or they belong to the signal mediated diversion to secretory vesicle. That means for the regulated secretion. So in the cell, some type of molecule they are constitutively secreted from the cell, and the other molecule they also be secreted from the cell, but they depend on the signal the cell receive. That means they not always be secreted. They can concentrate on the vesicle and wait until the cell receive the signal that at this moment the protein can be transported to the cell membrane, to the plasma membrane, and secreted. So, or the protein or molecule can be sent to the lysosome through the signal mediated diversion pathway. So, on this phase, on the transfer of the Golgi apparatus, the protein will be sorted to one among three pathway of the transport. The endocytosis from the plasma membrane also use the vesicular transport, so the cell can take up uh, a huge amount of molecule from the extracellular environment by the endocytosis. So clathrin mediated endocytosis on the plasma membrane is taken by the clathrin coating. So this is a image I already showed you how the clathrin coated vesicle is formed on the plasma membrane and transport all the molecule from extracellular to come into the cell. So for the endocytosis, sometimes they also need the trigger by the binding of the cargo to their receptor on the plasma membrane, and also in the other vesicular transport, you can see the interaction between the cargo and the receptor will trigger the vesicular transport. So what is the fate of the receptor for the endocytosis? That means after the endocytosis happen, so what will be the receptor, membrane receptor protein? So for now, there are three phases of the transmembrane receptor protein. They can be recycled. They can be recycled. That means after they do the transport function, so the membrane receptor can be sent back to the donor compartment, for example, like plasma membrane. to reuse. So that is the first phase of the transmembrane receptor. The second phase is the transcytosis. That means the localization of the transmembrane receptor protein will be changed after they do the endocytosis. For example, in this case, you can see the the transmembrane receptor is spread on the basal domain and then train to the apical domain of the plasma membrane. And the third phase of the transmembrane receptor is degradation. That means the receptor is degraded in the lysosome and there is no more receptor and the cell need to do a synthesize, the new synthesize to make a transmembrane receptor protein. So here is one example of recycling plasma membrane receptor. So this is a sample of the transport of the LDL molecule. So LDL is stand for low density lipoprotein. Low density lipoprotein, LDL. So this LDL is responsible for the transport of cholesterol into the cell. Because cholesterol is a lipid amphipathic molecule, so they cannot freely stay inside the cell or inside in the blood vessel. So they need the protect from the LDL. Then you can understand like the LDL, they make like the case, and all the cholesterol stay inside the case, and the LDL will be transported on the blood vessel to every cell in the body. So when the LDL stay in the extracellular environment, this LDL they can bind to their receptor on the cell membrane, on the plasma membrane. It's like in every cell in the body, they express the LDL receptor because, as you know, the cholesterol is very important for the plasma membrane, and the cell cannot synthesize this molecule, so they need import this molecule from outside. So they express the LDL receptor on their membrane. So LDL bind to their receptor and trigger the formation of the vesicle by clathrin coat. After the vesicle is formed and pinched off from the plasma membrane, so this vesicle will fuse with the endosome. After the fusion, because of the change in the pH, so the link between the LDL and their receptor is broken and release the LDL in the lumen of the endosome, and the receptor stay on the membrane of the endosome now is transport back to the plasma membrane and ready for another transport of the LDL. And for the LDL molecule, stay in the early endosome and then develop into the lysosome, and they can be degraded or they can be digested by the hydrolysis in the lysosome and uh, release the free cholesterol into the cell, and now the cell can use this cholesterol to make the new plasma or to make the steroid hormone if this cell is belong to the reproductive gland. So this is an example of the recycling plasma membrane receptor for the LDL transport uh, through the endocytosis. So you see, LDL can be transported by the receptor, um, by the binding of this molecule to their corresponding receptor on the plasma membrane. For the example of transcytosis transport, that means the change in the localization of the receptor. So in this case, it is example of the transport of the Fc receptor. So Fc is one part of the antibody. Uh, I don't know if you remember about the antibody. So the antibody, it has the Y shape with the Fc. That means the constant, and the Fv it means the variant domain of the protein. So all the antibodies they have their receptor on the cell membrane. So this receptor they bind to the Fc part of the antibody. So in this case, in the apical domain of the plasma membrane, if there is some antibody, so they can bind to the receptor through the Fc domain to the Fc receptor on the cell membrane and trigger the formation of vesicle to come and fuse with the endosome. So the fuse with the early endosome also in the endosome the binding of the receptor and the cargo stigmental. So they still stay on the membrane of the endosome. After the endosome continue to move into the in the cytoplasm. So one part of the endosome is from the transport vesicle contain the receptor and the antibody, and this transport vesicle continue to fuse with another endosome in the way to transport from the apical to the basolateral domain until the vesicle meets the basolateral domain of the plasma membrane and fuse with this membrane. So after the fusing, they can release the antibody in the um, basolateral domain of the plasma membrane, and also the receptor still stay on the plasma membrane, and they can ready to transport another antibody if they meet. And the third example about the degradation of endocytosine protein in the lysosome. So for the case of the degradation of the receptor, after the vesicle fuse with the endosome, so this endosome develop into the they can develop into the with the lysosome and form the structure of the lysosome together with the endosome, and they use the enzyme from the lysosome to cut all the vesicular in the consists of both the receptor and the cargo. So both receptor and cargo will be cut into in the lysosome, and after they can release some molecule that the cell needed, and for the receptor there is no more receptor. And if the cell need this receptor, they need to do the new transcription and translation to produce a new protein. Also, the other material imported into the cell will be destined to the degradation with the different pathway. So the endocytosis, as I already mentioned before, and some endocytosis after they take up the molecule from outside of the cell, and fused with the endosome and then the lysosome, and in the lysosome they will be hydrolyzed by the enzyme. Or if the cell do the phagocytosis, that means they can eat a very big part of the solid food from outside. So they take up by making the phagosome, and this phagosome can fuse with the lysosome. The other way is autophagy. That means the structure, some structure or some organelle inside the cell can be degraded by the formation of the autophagosome. That means they use a structure to surround the organelle and structure in the cell and fuse them with the lysosome and do the autophagy. Here is the image illustrate about the phagocytosis. Okay, so this process of phagocytosis is one example of phagocytosis. This is a bacteria. So the bacterium is it is eaten by one immune cell. So the immune cell they develop the structure they call the pseudopod and surround the bacteria, and then they will make the phagosome to fuse with the lysosome and degrade all of the bacteria inside the cell. This is a scan image of the other immune cell. They are eating the two spore, bacteria spore. Then you can see the two spore is eaten uh, by the cell. The way that they can eat the spore is they develop the membrane as a pseudopod surrounding the spore and then put the spore in the cytoplasm and then fuse with the lysosome to degrade this structure. I see. Okay. Okay, so the last part is about the autophagy. So as I say, autophagy is quite a new process discovered in the middle of the last century. And now we understand more about the autophagy in the way that the cell eat some of the structure or organelle. So autophagy, that means the self-eating. It is a lysosomal degradation pathway that is essential for the survival, differentiation, development, and homeostasis of the cell and the body. And in fact, the autophagy principally to protect the organism against diverse pathologies, including the infection, cancer, neurodegeneration, aging, and heart disease. So you see many important processes and also disease involving in the autophagy in the cell. For the first autophagy is identified in the red liver cell. So the scientists, when they do research on the liver cell, they found out that um, in the red liver cell they can eat or destroy many of their organelle, and in the stress condition. So the stress condition here, like the starvation, the hypoxia, or the heat stress. So whenever the cell face to the stress condition, they can train the number of their organelle or number of their structure by the autophagy process. And why they do that and how they can do that? Autophagy, it depends on the status of the cell. For example, if the cell suffer from the starvation, that means they have not enough the nutrient for their life, so they can reduce the number of the organelle in order to save the nutrient and the energy. So to survive in this condition, is this stress condition, that is one reason for the autophagy. Or in some stress condition like the heat, the increase of the temperature, so some protein is defect, or some organelle is mistake, is defect also, and the cell cannot repair. So in this way, in this case, the cell will destroy the structure or the organelle in order to maintain the stable of the cell. So that is some purpose of the autophagy. And how the autophagy can take place inside the cell. For now, the scientists identify about 32 different autophagy related genes. They call the ATG. That means autophagy related gene. And all of gene is identified in each. How the autophagy is regulated and executed at the molecular level have been made in each cell. So from each cell, the scientists figure out the 32 ATG gene involving in the autophagy. Um, and many of these genes are converted in the slim mode, plants, flies, and mammal. So you see, any gene which are which is conserved, sorry, which is conserved, that means they are important gene. So if you work with some gene that's similar in the human and the other species, like the plants, yeast, or bacteria, so this gene must be very important gene. So autophagy gene also belong to the important gene for the cell, and they are conserved in many species. There are three types of the autophagy: the macroautophagy, microautophagy, and the chaperone-mediated autophagy. So even there are three types, there is general feature for all of them. The proteolytic degradation of cytosolic component as the lysosome. So even they are macro or micro or chaperone autophagy, all of them involving in the lysosome. For the microautophagy, the structure of microautophagy is formed by the invagination of the lysosome membrane. The cytosolic components are directly taken up by the lysosome itself through, and it could be the selective or non-selective. So in this type of the autophagy, the membrane of the lysosome, they develop and they bound the molecule or the other component in the cytosolic and put into the lysosome and makes the digest. So the component can be the protein, lipase, or organelle themselves, can be digested directly by the lysosome. The chaperone-mediated autophagy also somehow similar to the microautophagy, but in the selective way. So the targeted protein are translocated across the lysosomal membrane in a complex with chaperone protein, such as the HSC70. And these complexes are recognized by the lysosomal membrane receptor, uh, LAMP2A. That means the lysosomal associated membrane protein 2A. So these complexes stay on the membrane of the lysosome and they can recognize the HSC70 So whenever they recognize this protein complex, they will they will push all of the complex into the lysosome, resulting in their unfolding and degradation. So because the structure or the cytosolic component, they need to bind to the HSC70, one type of the chaperone family, to send to the lysosome. So this they call the chaperone mediated autophagy. So chaperone here is one type of the protein bind to the degraded protein which sent to the lysosome. And the last, also the most popular autophagy is macroautophagy. So the macroautophagy is the deliver cytoplasmic cargo to the cytosomes through the autophagosome formation. So they will make the structure called the autophagosome, and this autophagosome will fuse with the lysosome to form an autolysosome and do the degradation of all the structure inside the autolysosome. And also it could be selective or non-selective, depend on the structure and molecule that they need to be degraded. So for the macroautophagy, they are the most popular, and also the most important type of the autophagy. And somehow when we say about the autophagy, that means we mention about the macroautophagy. So one macroautophagy process needs a three step. So the first step is the initiation, or the first stage is initiation stage. So in the initiation stage, um, they start from a phagophore. That means a lipid bilayer isolated membrane. So here in this image, you see, for example, this cytosolic component need to be degraded. So they will as a membrane from the ear or the Golgi apparatus or endosome will develop and surround the component here. They call the omegasome first. So that is the first step when the membrane plus the membrane developed from the ER or Golgi or endosome surround the cytosolic component to make the omegasome. The next stage is the elongation. So this phagophore expand to engulf intracellular cargo such as the protein aggregate or the organelle, ribosome, mitochondria, thereby sequestering the cargo into a double membrane autophagosome. So in the second stage, the elongation, they will make the autophagosome. So here, from the omegasome, this structure developed more by the development of the membrane to engulf the other cargo to make the autophagosome. So the small molecule or either the organelle like mitochondria can be contained in the autophagosome. And the last stage is a degradation, maturation and degradation. So the autophagosome mature and fuse with the lysosome to promote the degradation of autophagosomal contents by the lysosomal acid protease. So in this, after the autophagosome is formed from the state of the elongation, so this autophagosome will fuse with the lysosome to make the autolysosome, and in the autolysosome, the enzyme will degrade all the structure inside. So this is the step, on three step of the autophagy, the macroautophagy. And one thing I would remind us that after the degradation, many material can be released to the cell to reuse. So by this way, the cell can save energy and material for their life. Okay, so that also the end slide of the lesson today. And once again, I